Okay. Okay. All right. Okay, so I think we're going to go ahead and get started now. Is everyone able to hear me okay? Yeah, okay, sweet. And then is everyone able to see the PowerPoint as well? Perfect, okay, sweet. All right, so we're gonna go ahead and get into lab today. Actually, I think I have this is the wrong numbered lab. So this is lab 8, not lab 6. And this is gonna be on mitosis. So basically from here on out for the rest of the semester, we're going to be focusing on DNA and genetics. And so this is kind of where we start to shift gears away from proteins and carbohydrates and lipids, and we start to look more at DNA and genetic material and look at how our organisms as a whole are going to be able to divide and how they're going to be able to reproduce in a couple different ways, and then how that's going to impact various populations going forward. So I think it's going to get a lot more interesting kind of going forward here. So when we're thinking about cellular reproduction, there's different aspects of reproduction. So we can have reproduction of our own body cells, so helping our bodies grow, and then we can have reproduction in order to help us produce some offspring. Okay, so for today, we are going to be focusing on reproduction of our own body cells. Okay, so that's going to be through this whole process called mitosis. So when we think about mitosis, mitosis is a very important process because this is a process that allows our body to be able to allow our cells to divide. And so if you think about, the size you were when you were first born, probably maybe a foot and a half long, maybe if that. And then eventually you end up growing to be, a 5-6 foot individual. Again, you're no longer the same size you once were. Okay, the reason you're able to grow is because of mitosis, this process of allowing your body to undergo division, cell division. Okay. The same thing happens whenever you get a cut or you need to repair some part of your body. So if you get a cut, for example, your body's going to start bleeding, okay, because you have a physical opening inside your skin. Okay? In order to repair that, your cells are going to start to divide to help close up that wound. Okay, so mitosis is going to be a really important process to allow our bodies to not just grow, but also be able to repair itself. Okay, and so the way this whole process is going to be working is we're going to be starting with our cells in an interface, which is where our cells are just doing their normal daily activities, and then they're going to begin to divide. And so here we see some of these chromosomes, which are some genetic material, and we'll talk about these in lecture on Thursday. Okay. They're going to start going through this process of mitosis where they're going to undergo prophase, metaphase, anaphase, and telophase. And as we can see, we're going to end up with having cells that are essentially almost completely separated at the end of telophase. Okay. And then at the end of this whole process, we're going to end up dividing these cells in half. So we end up with 2 daughter cells originating from one original parent cell. Okay. So that's going to be the goal of today. That's going to be what we're looking at. Okay. There's also a second process, which we'll also be looking at in a little bit less detail, but we talk about that more probably in a week or two, and that is meiosis. Okay. So meiosis, we'll look at a video of that later today, but meiosis is a process of allowing our bodies to produce genetically different offspring. Okay, so allow us to be able to produce offspring that are a little bit different from one another. Okay. And so again, we'll take a look at those in a little more detail going forward here. Okay. But for this week, we're going to be focusing mainly on mitosis. Okay. So mitosis, or the mitotic phase, is going to be a process where we're going to be taking one cell that has its own nucleus and its own cytoplasm, and we want to split it in half, okay, and end up forming 2 new daughter cells at the end of that process. Okay. So in order to do that, we're going to have to go through 2 different types of activities here, 2 different processes. The first one is mitosis, where we're going to be taking our chromosomes, which are our DNA that's been condensed into a smaller form. And we're going to take our chromosomes and we're going to line them, separate them, and then move them to opposite ends of the cell. Okay? And then we're also going to be dividing our nucleus. Okay? So the process of mitosis is divided up into 5 stages, sometimes 4, depending on who you're asking. But there's going to be 5 stages where we're going to have prophase, we're going to have prometaphase, we're going to have metaphase, anaphase, and then telophase. Okay. At the end of telophase, our cells are not fully divided yet. And so we have to go through a second process called cytokinesis. And so cytokinesis is what actually allows our cells to physically separate from one another and become 2 independent daughter cells. Okay. So we're going to take a look at how this process works. And again, if this doesn't make a whole lot of sense, don't worry about it. We'll talk about it in more detail on Thursday when we have a lecture. Okay. So the first phase is prophase. And so in order to start undergoing cell division, the first thing that we need to do is we need to make sure that our nucleus is as easily accessible as possible. Okay. So what we're going to be doing is we're going to get rid of the nuclear envelope. Okay, so we're going to have this nucleus that's going to have a double membrane surrounding it. Okay. We want to get rid of that nuclear envelope and break it down so that we can easily access the contents of the nucleus, access the DNA inside the nucleus there. Okay. That's going to be the first that we need to do. Okay, break down the nuclear envelope. Okay. The next thing that we're going to do is we're going to take the Golgi apparatus and the endoplasmic reticulum that are all going to be surrounding the nucleus as well, and we want to break those components down as well. Okay, so here we have our rough ER, here we have our Golgi. We want to get rid of those and break those down, and then move those components to the periphery of the cell. Okay, so now that they're kind of out of the way, so again, we can now access the nucleus a lot easier. And then the third thing that's going to happen is there's going to be the nucleolus, which is that little ball inside the nucleus. Okay. We're going to cause the nucleolus to disappear at this point as well. Okay. To finish off prophase, we're going to start producing some microtubules from these organelles called our centrosomes. Okay. So our centrosomes up here, these 2 kind of cylindrical looking guys, are going to start producing these microtubules, which we'll call mitotic spindles. And those mitotic spindles are going to end up pushing the centrosomes to opposite poles of the cell. Okay, so one's going to end up on one side, the other set is going to end up on the other side, okay, with the mitotic spindles kind of branching off of those guys. And we'll see where these guys come into play in a second here. Okay, and then we're going to have our sister chromatids becoming more visible. Okay, so when we look at our chromosome, here we go. Our chromosome is going to kind of look something like, let me do this in a different color. Our chromosomes are going to look something like this. Okay. So this whole thing would be 1 chromosome. I'll just put a C cell. 1 chromosome there. Now, each of these little knobs at each end here, okay, is 1 sister chromatid. I'll put just C tid. Okay, so this would be 1 sister chromatid, and this would be the other. Okay, all of this would be 1. This part would be the second sister chromatid. Okay, so in 1 single chromosome, in this case, we're going to be looking at 2 sister chromatids making up 1 chromosome. Okay, is everyone kind of following me so far? It's all kind of making sense. Yeah, okay. All right, so that is prophase. Okay, so I'll go ahead and just, okay, so that's prophase. So again, all you're doing here is we're breaking out the nuclear envelope, we're getting rid of the Golgi apparatus and the endoplasmic reticulum and moving them to the periphery of the cell. We're getting rid of the nucleolus. We're going to start producing our microtubules from our centrosomes here, and we're going to start moving our centrosomes to the poles of these cells. So that's the end of prophase. Okay. The next phase, which depending on who you ask, some scientists or some biologists will consider prometaphase a phase, some will just not really consider it a phase. So depending on who you ask, you may either consider it, you may not. For our case, we'll just consider it for now. But essentially, prometaphase is just an extension of prophase. Okay, just basically the last half of prophase. Okay. So what's going to be happening in prometaphase is the nuclear envelope is going to finish disappearing. It's going to finish breaking down. Okay. And then the microtubules are going to fully push all of your centrosomes to the opposite ends of the cells. Okay. And then what's going to happen is those microtubules are going to begin to latch onto each of the chromosomes at a particular region called the kinetochore. Okay. So this kinetochore is just a protein that's going to attach to the mitotic spindles. Okay. So yeah, so Johanna's question was if this part is in activity 1. You don't need to write this part down for activity 1. Okay, I'm just kind of going over it just for the sake of going over it, but for our cases, we're not going to worry too much about this. Okay, so that's the mitotic spindles. So the mitotic spindles are going to attach to the chromosomes at the kinetochore. And every sister chromatid will have a kinetochore where the mitotic spindle will attach. Okay, that's prometaphase. Okay, so the next phase here is metaphase. Okay. And so metaphase is going to be where our chromosomes are going to begin aligning along the metaphase plate. Okay. So at this point, our sister chromatids, so our chromosomes have essentially attached to the mitotic spindles. Okay, at this point, the mitotic spindles are going to arrange the chromosomes in a single file line along the metaphase plate, which is basically at the equator of your cell. Okay. So they're going to line them up in a single file line here. And your sister chromatids, like I said, are still going to be attached to the chromosomes, to one another, and they're still going to be attached to the mitotic spindles as well. Okay. And at this point, our chromosomes are going to be fully condensed, and we'll talk about what that means on Thursday in lecture. Okay. So they're going to be lined up right along the metaphase plate. Okay, down here, I have a picture of some fluorescence microscopy of having some of a cell in metaphase. So these blue parts right here, these are the chromosomes, okay? Again, all aligning at the metaphase plate. All these green components here are the mitotic spindles, these microtubules that will be attaching to those chromosomes. All right, cool. So that is metaphase, okay? Again, the only thing that's happening in metaphase is that they're just gonna be lining up along the metaphase plate. Okay. The third, or I guess fourth phase of mitosis is anaphase, okay? So in anaphase, what's going to be happening is we're going to be separating the sister chromatids from one another. Okay. So again, remember, each chromosome is going to have one sister chromatid, or is going to have two sister chromatids. We're going to have one on this side, and then this other leg is going to be the other set of sister chromatids. Okay. So in metaphase, they're all going to be aligned at the metaphase plate. Okay. In anaphase, you're going to be separating each of those chromosomes and pulling them completely apart. And you're going to be moving them to opposite ends of these cells towards these centrosomes. Okay. So at this point, now that we've separated all of our sister chromatids, now each sister chromatid is now considered an individual chromosome. Okay. So now this is one chromosome, 2, 3, 4, 5, 6, 7, 8 chromosomes now, as opposed to the 4 that we started off with during this process here. And so during this process, we're going to have our cell elongating at the same time. Okay. So again, if we look at our fluorescence microscopy here, we can see our chromosomes now being pulled apart to opposite ends of these cells. All right. Then our last phase is going to be telophase. Okay. And so in telophase, the cell is getting ready to split into 2 individual daughter cells. Okay. And so what we're going to be seeing here at the end of this whole process is we're going to end up seeing 2 nuclei starting to reform inside of a single cell. Okay, so the way this is going to work is those chromosomes that have just been pulled to opposite ends of the cell are now going to begin to decondense. And again, we'll talk about what that means on Thursday in lecture. Okay. So they're going to be moved to opposite ends of the cell, and they're going to start decondensing. Okay. The mitotic spindles that we initially had now or before are going to release from the chromosomes, and they're going to start to break down. Okay. And then we're going to reform our nuclear envelopes, just like we see over here. Okay. So these are our nuclear envelopes beginning to reform. Okay. So you can kind of think of telophase as the opposite of prophase. Okay. Basically, in telophase, you're just undoing whatever you did in prophase here. Okay. So again, if we look at our fluorescence microscopy over here, we're going to see a group of chromosomes on this side. We're going to see a group of chromosomes on this side, and we're going to see our cell beginning to pinch off, okay, here in the middle. Okay. So here's the pinching region over here. Okay. So again, we still have all of this within one single cell, but we have two nuclei in here. Okay. Everyone following me so far? Are there any questions before we move forward here? Yeah, this is pretty cool. It's pretty cool. Okay, so with that, now we go into this last phase. Okay, so at this point, we have formed one daughter cell that has two nuclei in it. Okay. Now, that's not really a situation we want to have. So we want to end up splitting that cell in two. Okay. So that last phase where we're going to be splitting our cell in two is going to be this phase called cytokinesis. Okay. So in cytokinesis, this is where we're actually going to physically separate all the cytoplasm and the nuclei into two individual daughter cells. Okay. Now, depending on the type of cell that you're looking at, this is going to work a little bit differently. Okay. So it's going to be looking differently for cells that have a cell wall versus ones that don't. Okay. And so we're going to start by looking at animal cells which do not have a cell wall. Okay. So for animal cells, cytokinesis is going to start right around the beginning of anaphase. Okay. And so what's going to be happening is we're going to start forming our actin filaments. Okay. So if you remember from the earlier parts of chapter 3, there were 3 different types of fibers that are, or aspects of the cytoskeleton that our cells are going to be producing. And one of those is actin. Okay. So actin, or these actin filaments, are going to start wrapping around the metaphase plate, okay, right around here. Okay. Once they start forming around the metaphase plate, that actin is going to start to constrict around that particular region and form a little fissure called the cleavage furrow, okay, which we see over here. Okay. And so as that cleavage furrow, as that actin begins to constrict more and more and more, it's going to constrict until you end up pinching your cell into two individual daughter cells. Okay. And so we're going to start off with having a full cell here. We're going to have our actin filaments beginning to pinch until you end up forming two new daughter cells at the end of this whole process. Okay. So we started with one, and now we ended up with two. Okay. So that is how cells are going to be able to divide in animal cells. Okay. In plant cells, this process is going to be a little bit more complicated because of the cell wall. Okay. Now, the cell wall is very strong and it's a very rigid structure. So adding actin filaments to try to pinch around it wouldn't work because the cell wall is too strong. Okay. So instead of trying to pinch the cell and breaking it into two, what we're going to do instead is we're going to build a new cell wall in between the two nuclei. Okay. And that's going to help to form the two new daughter cells. Okay. So the way this is going to work is we're going to kind of go back to interphase, which we haven't really talked about, and we'll talk about that more on Thursday. But at the beginning of interphase, which is where the cell is starting to prepare to undergo division, what's going to happen is the Golgi apparatus is going to start to accumulate a bunch of different stuff. Okay, so it's going to take in some enzymes, it's going to take in some structural proteins, and it's going to take in some glucose. And it's going to store them inside the vesicles within the Golgi apparatus. Okay. Now, when prophase hits, the Golgi apparatus is going to start to break apart, and it's going to move all those vesicles to the outside of the cell. Okay. So those vesicles are going to be on the outside of the cell, and they're going to remain there throughout prophase, metaphase, anaphase, and telophase. Okay. Now, when it's ready for cytokinesis, when it's ready to actually come back together, what's going to happen is those vesicles, those Golgi vesicles that were containing the structural proteins, the enzymes, and glucose, are going to start to come back along the metaphase plate. Okay, they're going to reorient themselves at the metaphase plate and line up together right there. Okay. So eventually those vesicles are going to start fusing together with one another, and those structural proteins, the enzymes and glucose, are going to all kind of interact with one another, and they're going to end up fusing. And what's going to happen is you're going to end up forming a cell plate in between the two nuclei. Okay. Eventually that cell plate is going to continue to enlarge and enlarge and enlarge, and eventually it's going to fuse with the existing cell wall and then form, a fourth wall in between these two cells. Okay. And so that is going to be how you end up forming two new daughter cells in plants, as opposed to having just the one. Okay. And so the glucose that we accumulated before ends up forming the cellulose that is that makes up the cell wall of our plant cells here. Okay. So this process is a little bit more complicated in plants versus in animals here. Okay. So Johanna's question, yeah. So Johanna's question is: sister chromatids separate in anaphase? Yes. So in anaphase, those sister chromatids are going to separate from one another. Okay? So again, here we have one sister chromatid on one side and then the other sister chromatid on the other. Okay? In metaphase, okay, in anaphase, you're going to actually physically separate those sister chromatids from each other. And then once they separate, they are now considered an individual chromosome. Okay, cool. Good question. Any other questions here? No. Okay. So if there's no more questions, what I'll do is I'm going to have us watch a couple videos, watch one video on mitosis. And so hopefully you guys are going to be able to hear this. So here we go here. Okay, so this is going to be a quick video on mitosis. Okay, and let me know if you guys are able to hear this okay. Ooh. Oh no, okay. Okay. Okay, so you guys can't hear it. Okay, give me 1 second, see if I can figure out.
We can.
Yeah. Okay. So yeah, it looks like the audio is not playing all that well, so I'll just kind of leave it alone. That the links are on the lab page assignments that I've uploaded onto Canvas, so you can watch those later, and hopefully you'll be able to hear from your own computers when you watch it there. But I want you to watch this video on mitosis, which again kind of just helps to kind of easily kind of show you exactly how mitosis is going to be working here. So I want you to watch that video, and again, we'll talk about it in more detail when we talk about it in lecture here. Okay. Okay, is everyone able to see the PowerPoint again? Yeah, okay. Okay, so that's mitosis. Meiosis is going to work very similarly. And again, we'll talk about that in chapter 7, so don't worry too much about it now. But I do want you to watch that video, the meiosis video as well, because one of the questions at the end of the lab has to do with comparing mitosis versus meiosis. So make sure you watch both of those videos, and it'll kind of help you to kind of clarify what's going on in all of these phases here. Okay, so as you watch those videos, what you're going to do is you're going to be looking at that table. I'm going to kind of go back to the lab assignment page. Okay, and so watch those videos, and then when you watch those videos, you're going to be able to draw in what's going to be happening in each phase of in each of these phases here. Okay, so again, each of these circles represents one cell. Okay, so you can just draw in what's happening with the chromosomes in each of these phases here. Okay, so you can go ahead and skip prometaphase because it's really not that crucial. Go ahead and skip prometaphase, but make sure you do all the other ones. And you can go based off of the video to kind of help explain what's going on. For before DNA, do we draw 2 chromosomes? So when you watch the videos, it'll kind of help you to see what you should be drawing. Yep. So watch those videos and it'll show you what you should draw for before DNA duplication. But essentially, you won't be able to see those two chromosomes. It'll basically be looking like little squiggly lines. Okay. But if you watch the videos, it'll show you. It'll show you in pretty good detail. Okay, so that'll be for activity 1. Okay, kind of going back to the PowerPoint here. Okay, resume slideshow. Okay. Okay, actually, let me backtrack. So there's an easy way to kind of help you learn prophase, and so I learned this back in high school, and I never really forgot it. The way you kind of help you to kind of help yourself remember all the different steps of mitosis is to use your hands. Okay? And so if you take your hands, you take your two hands, your fingertips represent your chromosomes. Okay? Your fingers will represent your mitotic spindles. Okay? So the first that's going to be happening is prophase. Okay? So in prophase, our chromosomes are just kind of all over the place. Our mitotic spindles are beginning to form here. Okay? Nothing too crazy is going to be happening. Okay? But everything is just kind of all over the place. Okay. The next phase is metaphase. Okay. And so in metaphase, your chromosomes, your fingertips here, are going to line up along your metaphase plate. Okay. Then we get to anaphase, where we physically separate our sister chromatids. So our chromosomes are now separated here. Okay. In telophase, we start to reform our nuclei, which are going to contain our chromosomes. And then the last phase is cytokinesis, okay, where you actually physically move and separate your two cells into two components. Okay. There are two daughter cells here. Okay. So again, we got prophase, chromosomes all over the place. Metaphase, where we have our chromosomes lined up at the metaphase plate. Anaphase, where we separate our chromosomes. Telophase, where we start to reform our nuclei. And then cytokinesis, where we form our two new daughter cells. Okay. So that's an easy way to kind of help you remember the phases of mitosis. Cool. Yeah, pretty smart. And so, like I said, I learned that back in high school and never forgot it. So when exam 3 comes around, if you need to start using your hands to try to figure out what steps of mitosis you need to do, feel free to do that. Okay. Okay. So that is mitosis. Okay, so that's that. So activity 2, we're going to skip, so just don't worry too much about that. Activity 3, what we're going to be doing is we're going to be looking at various types of whitefish cells as well as some onion root cells. Okay. And so these cells are going to be shown in the lab assignment as well. Okay. Your job is to count the number of cells that you're able to see in interphase, prophase, metaphase, anaphase, and telophase. And see if you can count a total of 50 cells or just count however much you're able to see in each of the pictures. And then calculate the percentage of cells in each of those stages. Okay. So I'll kind of show you a few examples here. So if we look at this top left picture, okay, we're going to see a few cells in that are undergoing mitosis. Okay. So if we look at this set of cells here, okay, or this particular cell here, we're going to see our chromosomes, which are these darker structures right here, okay, the guys outlined in black. Okay. Here we see our chromosomes all lined up in a single file line. So what phase of mitosis are we looking at? Is this prophase, anaphase, or prophase, metaphase, anaphase, or telophase? Right, so everyone's saying metaphase. Right, so this is exactly right. So this would be metaphase here. Okay. Now, if we looked at this cell over here, okay, if we're looking at this cell, if we start to see our cell starting to pinch off here, okay, and we start to see groups of chromosomes just along, kind of on their own here. So which phase of mitosis do we think we're looking at with this cell here? Right. So Johanna and Jared got this one right. So this is telophase. Okay. Telophase and anaphase are a little bit difficult to kind of differentiate, but this would be telophase. And one of the ways that you're able to tell is there's not a whole lot of very thick lines in between the two of them. Okay. So this would be telophase. Okay. Here, we would be seeing anaphase. So you can still see this darker structure around behind our chromosomes. Okay? And so here we have our chromosomes. You can see them being pulled apart. Okay? You start to see some darker lines in between, which starts to kind of help us see that there's still some chromosomes in the middle, or our sister chromosomes in the middle here getting fully separated. So this would be anaphase. This would be another anaphase cell over here as well. So those are some of those phases over here. Okay. If you take a look at these ones over here on the right, okay, again, here we'd be looking at anaphase. This one, what do we think we're looking at here? We have our chromosome just kind of all clumped up here. What do we think we're looking at? Prophase, metaphase, anaphase, or telophase? Yeah, so Johanna's got this one right. So this one would be prophase. Okay. So this would be prophase because we're only seeing a single cell, and we just see our chromosomes all just clumped up in a single area. Okay. There's no rhyme or reason to where they are. They're just all kind of clumped up in that particular region here. Okay. So that would be prophase. Okay. Again, here we kind of start to see our chromosomes lining up along a single file line. It's a little faint, but this would be metaphase here. This cell over here, I want to point out because this one we can't really see any chromosomes in it. Okay, so because we can't see any chromosomes in it, this shows us that our chromosomes have not condensed yet. Okay, and so this would be interphase. Okay, so this is interphase. This is before mitosis has begun. Okay, so this is interphase. Cool. So this would be interphase. This cell would be an interphase. This cell would be an interphase. And so the majority of the cells that we're going to be seeing are going to be an interphase because that's where our cells are going to be spending the majority of their time in. Okay. Okay. So these are just a few more examples. So you can kind of go through those if you need a little bit more practice. So I have a few examples that are also outlined in the lab assignment as well. So if we kind of go over here, I'll kind of zoom into this so you guys can see this a little bit easier. So here we are able to see some cells that are in different phases of mitosis. Okay, so here we've got a cell that's in anaphase. Here we've got a cell that's in metaphase, and metaphase over here. Here we see prophase. Here we see telophase. Okay, so these are just a few examples. Okay, the ones that you're going to, and then so what you're going to be doing is you're going to count the number of cells you're able to see, okay, in each phase. So let's say, for example, we were able to see 20 cells in interphase, 15 cells in prophase, 10 cells in metaphase, 12 in anaphase, and 8 in telophase. You're going to total up the number of cells that you're able to get, and then you're going to come up with a percentage. Okay? So the 20 out of 65 cells were in interphase, that's a total of about 31%. 15 out of 65 cells in prophase, that would be about 23%, and so on and so forth. Okay? So you should get about 100% over here. Okay? You're going to skip the number of cells in the group portion here. Okay? Just skip this row. Just worry about the number of cells individual and then the percentage in each stage. Okay? So you're going to do that for both the whitefish cells as well as the onion root cells. Okay? So the ones you're going to be looking at here are going to be these guys for the whitefish. Okay? And so again, you try to identify how many are in each phase: interphase, prophase, metaphase, anaphase, and telophase. Okay? And then write that into that table. And then you're going to do the same thing with these cells, or for the onion root cells. Okay. And again, so you're going to do the best you can. You don't need to get a like an exact number. I'm not grading you on if you're able to come up with the exact number, but this is just more for practice to be able to identify all the different phases of mitosis. Okay. And so you're going to write that in there. Okay. So these are just those cells here. And again, so you're going to write that in for the whitefish cells, and then you're going to write them in again here for the onion root cells. Okay. And then there's some space over here for you to answer the extra questions, which are on page 64 of your lab manual. Cool. Okay. Any questions on anything as far as what we're doing for this lab? No. Okay. So if there's no questions, I'll go ahead and just stop this meeting here. Go ahead and complete this whenever you get a chance. Make sure that you submit this by 11:59 p.m. Ideally, if it takes a little longer and you have to get to it by the end of the week, that's fine, because I know this is a little bit complicated. But yeah, so the only activity that you're going to be skipping, so to answer Kimberly's question, the only activity you're going to be skipping is activity 2. Okay, so you're doing number 1 and then you're doing number 3. Okay, but you're skipping activity 2.